Okay guys, so I'm going to go over the second part of the DNA lab. So after PCR, if you had a question about PCR, I had a previous video on that. But again, this is a full picture of what we're looking at. We have a genotype, and remember, just in the cell, we go through transcription, make that mRNA, and go through translation to make a protein. And that protein could be functional, like for a taster, or a non-taster where it's non-functional. Okay, and then that results in a phenotype of being able to taste or not taste that PTC chemical. Okay, so you either taste it and it tastes kind of gross, or you don't really taste it. Okay, so again, our big overview is that we have DNA that we extracted from our cheek cells. We then did PCR to increase the number of copies of that piece of DNA, and then we did restriction digest, and then gel electrophoresis to visualize that. So we're emphasizing the second part. Okay, so restriction enzymes, these are just enzymes that look and scan through the DNA and they cut at a particular location. Here's two examples of restriction enzymes. This EcoR1 looks for GAATTC. Whenever it sees it, it cuts it at this particular location. Okay, here's another example. We have GCN. GCN here is representing any nucleotide, so it could be GCA, GC, GCT, GC, GC, C, GC. It doesn't matter what that nucleotide is. Just as long as it says GC something, GC, it's going to cut right there. Okay, so we can use this and add it to our DNA, and it will identify the sequence. Here we see GCA, GC, which is the same as this. And this enzyme will come through and cut that DNA into different pieces. Okay, so that FNU4HI is the enzyme that we used in our particular lab. There's lots of different enzymes that recognize different sequences. But how it works is if you are a taster, so you were able to taste, you have a functional protein that is produced, this is the sequence. And if you are a non-taster, this is the sequence. So notice that this C was had a substitution mutation to be an A, and it changes our sequence. So here we see GCT, GC, which would be recognized by our restriction enzyme, but GTTGC is no longer recognized. So that means if we have a taster allele, it is going to be cut, and we will see two different DNA. The DNA will be cut into two pieces. When a non-taster, it is not going to be cut. Okay, that's while we're exploiting this sequence that this particular enzyme uses. Okay, so remember that we have lots and lots of pieces of DNA from our PCR from last class, and that PCR is kind of like taking that gene that's really, really long and multiplying it over and over and ending up with a stack of papers where we've copied it 63 miles high. Okay, so we have lots and lots and lots of this DNA. But now we need to cut it. Okay, so we're just adding literally our PCR product, that DNA from last class, and that enzyme, and we put them into the tube. We put the restriction enzyme here represented by these scissors, and we're just going to have it incubate there at 37 degrees, which is body temperature. So the temperature the enzyme is happy at, so it can do its thing. And it goes through again, cutting our taster DNA, but not our non-taster DNA. Okay, in class we then tasted the PTC to see what our phenotype is. And then after that, now we have cut our DNA, but now it's still in a tube. We still have a tube of DNA, and cut DNA or uncut DNA still looks like a tube. So we need to be able to visualize and see, well, did it cut or did it not cut? And that's the purpose of our gel electrophoresis. So our gel electrophoresis, how it works is we make a agarose gel. It's kind of like a seaweed compound, but agarose is like a jello. So it's like a jello little rectangle. It's got little holes here we call wells, and we put it in our gel electrophoresis machine and run a current through it. And we have a negative end and a positive end. Okay, how it works is DNA here, represented by these purple circles, is negatively charged. Okay, so it's going to be attracted to this positive end of the gel. So once we have the electrical current going through, those negatives are going to start moving towards that positive side. But larger molecules, like our larger molecules of DNA, have a harder time going through that gel than smaller molecules. Okay, so we're going to use this to separate out to see if we have cut or uncut DNA. If we have uncut DNA, it's going to be a longer piece of DNA, a longer, larger molecule. It's going to get trapped up more and go slower. If it's a smaller molecule, like that cut fragment or piece of DNA, it's going to move faster through the DNA or through the gel. Okay, so we pipetted and loaded our gel, meaning we just put the DNA sample into our electrophoresis gel, and then we ran an electrical current through it. This is some information about how we made the gel. I have another video about how to make the electrophoresis gel if you're interested. Okay, so here is an example of some possible results. We might get one band here. Okay, if we have one band, that tells us that the DNA was not cut. Okay, the DNA size, it was not cut. And if you remember, that means, if we go back, uncut DNA means it is a non-taster. Okay, so a non-taster here is going to be represented by this one line. Let's look at B and at C. And let me just take a snip of this really fast so I can draw on it a little bit. Because if we have uncut DNA, we are a non-taster. And that also is, to be a non-taster, you are going to be recessive. Okay, so I'll use the letter A. Sorry, I'm trying to use my mouse so it's not the best. So that's going to be the letter A. If we are a non-taster and all of it is uncut, that means we have two copies of that recessive non-taster DNA. B here, we have two bands. Okay, two bands, and they're both smaller than the previous band. So that's telling us we have two different bands, that we have one piece of DNA that was cut into two pieces of DNA. Okay, so that means we have a taster. Okay, and all of the DNA was cut, so we have a homozygous taster. The other option we can get is a little mixture between the two, where we have three bands, one larger band and two smaller bands. So, and notice they're the same size as the other bands. So that means we have some DNA that was cut and some DNA that was uncut. Now, this could happen because the enzyme wasn't working properly and didn't get to cut all of the DNA. Or it could be because some of the DNA or one copy of the DNA is dominant and one copy is recessive. These are kind of our three different options that we would expect from our PCR and our restriction enzyme results. Okay, this is what it would actually look like on a band. This is a mirror image. I should probably flip it in the future, but this is showing a DNA ladder. It's just used for size comparison. And then we have our bands here. So a single band would look something like this. Here's a double band where it's all cut. And then if we have three bands present, it looks something like that. Okay, we're using PTC in this example because it's simple. We actually only have one gene that's looking at this. There's other ones. Well, there's actually more genes affecting this particular allele. So it's a little bit of a messy experiment, but it's easier than some other genetic things we could be looking at. Okay, so that is my summary of what we did in our DNA lab. So make sure you go through and look on the Canvas assignment, and you can see our DNA results that we got, and use that information too, and it tells you whose results were where. So you can use that to predict what the genotype and phenotype of the individual is. We also have a chart that says who could taste and who could not taste. So you can fill in if we were correct in our assumption from our DNA analysis, or if our assumption was correct from our tasting. So we have two pieces of evidence, the physical tasting, and then that DNA analysis. So hope this is helpful. Bye, guys.